Log on, tune in, find out. Another good idea from Cambridge.
Thank you, Tandy. Good morning, everyone. Yeah, so I will be talking today about some of the recent work we have been doing on reconciling gene trees to infer a species phylogeny. And when I proposed the title and abstract, I was thinking about talking about very recent work on integrating lineage sorting and hybridization. But after the talks yesterday, I noticed that there wasn't much about the background for the gene tree, species tree. So I decided to put some slides on the background where there's no hybridization, it's just incomplete lineage sorting, and from there we will move on. Okay, so the talk will have basically three sections. The first one is some sort of background talking about the issue of coalescent histories. When we think about gene trees and species trees, what is a coalescent history? Because it is really the basis for some formulations for the reconciliation of gene trees. The second part, I will talk about some of the recent work we have done on a parsimonious way of reconciling gene trees and along with the algorithms that we have come up with. And the last part, I will focus on this issue of how do we deal with the case when gene trees may disagree because of hybridization and incomplete lineage sorting at the same time? How can we detect that? So we'll start with the first part here where I want to define this concept of coalescent histories. So the general setting for this problem is really looking at the species tree from a population perspective, where if we look at this species tree and we try to understand how to reconstruct this species tree, we need to keep in mind that this is really not a single line that we draw with the tree. It is really these tubes here within which we have a population of individuals growing. And in this species tree here, each row corresponds to the individuals at that generation, and you can go backward in time to the most recent common ancestor of all species. Now, when we look at the species tree in this context, then we can see that within that species tree there are other trees growing. Like one of them here is the green one, which is what we call the gene tree. Now, I want to emphasize that the concept of, or the notion of gene tree is really a historic name. It doesn't have to be a protein coding gene. It is the important thing to keep in mind here is that when we construct these gene trees and we want to infer species phylogenies, is that we really focus on genomic regions that do not have recombination. Okay? So it's basically the recombination-free regions from the genomes. It doesn't have to be, again, protein coding. Now, when we look at this in this context, now we start tracing the evolutionary history or the genealogy of these alleles that we sampled from the present day. Then we start going back and look at this gene genealogy. Then we might start seeing different histories than the species tree. So if we ignoring that fact that the gene trees might disagree with the species tree, basically we go to the pre-genomic era where to reconstruct the species phylogeny, basically one would focus on a certain locus in a set of species of interest like A through E, then sequence that locus, build the gene tree, and then the assumption is that the gene tree equals the species tree. In other words, once you build the gene tree, you will say, here's my species tree. However, in the post-genomic era, now that we have whole genome sequences, and in particular we have multi-locus data set, sometimes we don't have the whole genome, and we don't need the whole genome. We need the multiple loci. In this case, we have multiple loci to sample and sequence, and we can build the gene tree for each one of them. Now, once you build the gene trees and you start looking at them, if you look at this case here, for example, you will see that the gene trees do not agree with each other in terms of topology. So throughout my talk, I will not talk about coalescence times on the gene trees or branch length. So I will talk about topological discordance among the gene trees. Okay? Although it is very important to keep in mind that two gene tree topologies might agree, but the gene trees might give different signal because the branch lengths or the depth of these genealogies are different. Now, once we look at that, the problem becomes, let's get a species phylogeny. And the reason I'm not calling it a species tree because it does not have to be a tree. Species phylogeny might also have these horizontal edges that correspond to horizontal gene transfer or hybridization or recombination and so on. Okay, so the problem of interest for us is how do we go from these multiple loci to this species phylogeny. And my focus will be about going from the gene trees to this. The work I will be describing does not go directly from the sequences here. So we will assume that you have built the gene trees using your favorite method, likelihood, Bayesian approaches, parsimony, neighbor joining, whatever, and now you want to reconcile them into the species phylogeny. So just to talk about this concept of incomplete lineage sorting, I want to put back again that concept of the species tree and looking at the population. So you can see that within this species tree there is this red gene tree growing there. And if we trace that genealogy for the alleles or lineages that we sampled from the 3 species, we will notice that that gene tree does not give the same topology like the species tree. And if we look at this species tree, the main factor that will affect the probability or the chance of observing such a gene tree is really the length of the branch that separates human chimp from gorilla, which is the length of which is T2 minus T1. Okay. Now, this is not a new problem, actually. The probability of observing each of the 3 possible gene tree topologies have been described for over 3 decades probably now. So if you are given under neutral evolution and no population structure or gene flow or anything like that, if we have this species tree and I know the times T2 and T1, these 2 speciation times, then I can calculate the probability of each of the possible gene trees as follows. Okay, so these will be the 3 gene trees that are possible with their probabilities. If we want to visualize these probabilities, this is how they look. On the x-axis is basically the length of the branch that separates human chimp from gorilla, and on the y-axis we see the probability of each one of the 3 possible gene tree topologies. As the x-axis increases, you'll see that the yellow region becomes the dominant one in the sense that the probability of this gene tree starts getting closer and closer to 1. Okay, so if this branch is very long, the chances for incomplete lineage sorting, or the chances that you will observe gene trees that disagree with the species tree, start decreasing. Okay, and this is why if you are dealing with a data set where the branches are very long, it's usually very hard to invoke incomplete lineage sorting as a cause of incongruence. Now, central to reconciliation criteria that I will look at is the concept of coalescent history. So if given a species tree and a gene tree, then I can explain the gene tree by a pattern or by a sequence of coalescent events that could have taken place in the branches of the species tree. So to illustrate, suppose we have that species tree and we have this gene tree here. So they are agreeing. Now to give rise to a gene tree like this, if I sample an allele from A, an allele from B, an allele from C, and I start tracing their evolutionary history or genealogy backward in time, to give rise to this gene tree, if I ignore lengths of branches in the species tree and times, then there are 2 possible ways that coalescent events could have happened to give rise to this gene tree. One of them is for A and B to coalesce on this branch, and the other, which is this one, and the other one will be that A and B coalesce deeper than their MRCA. They coalesce here, and then they coalesce with C. Okay? There are no other options here. These are the 2 possible ones. Now, this is a very simple case. If I give you, you can easily come up with examples of a gene tree and species tree on 20 taxa where the number of possible coalescent histories is in the millions and tens of millions. Okay? So this number grows very, very fast. Okay? So this is the notion of coalescent histories, that if you are given a gene tree, you can put a sequence of coalescent events within the branches of the species tree so that you give rise to such a gene or explain such a gene tree. Now, if we denote by the set H sub ST of GT, the set of all coalescent histories of species tree, the set of all coalescent histories of gene tree, GT, and species tree, then this concept here of this set of coalescent histories is the basis for certain definitions for reconciling gene trees. Noah Rosenberg and our group in 2007, we gave algorithms for enumerating this set. Okay. So what do I mean by they give rise to the criteria for reconciling gene trees? So now, to reconcile gene trees in a species tree and in a probabilistic setting, we have to have a calculation of what is the probability of a gene tree What is the probability of observing gene tree GT given species tree topology and its branch lengths? Okay, and this formulation here is from a paper by James Degnan and Laura Naukobatko-Salter in 2005. And the definition of this formula is a summation over all valid or over all coalescent histories. Okay, so if you look at this, the probability of a gene tree given a species tree and branch length, it takes the summation over all coalescent histories. For each coalescent history, we do this calculation. Now ignore this term for the time being. Here we now take the product over all internal branches of the species tree. So go from 1 to n minus 2. This is the number of branches in the species tree, the number of internal branches. For each branch, we now, if you are given the coalescent history, you know how many coalescent events are taking place within each branch, and you know the order, the specific events. So now you have to calculate. So suppose you have within a branch 3 coalescent events. Now you have to ask, what is the probability that 3 coalescent events could have taken place within that amount of time, which is this term. So it says the probability of U lineages entering that branch, V lineages exiting that branch within that length of the branch, okay? And this term here is to give us the probability that, suppose now I know that 3 coalescent events have taken place. What is the probability that this specific set of 3 coalescent events have taken place? This is exactly the same like this, but this is just for a special branch that is over the root of the species tree. So there's always that branch over the root of the species tree where you have to take this probability. The probability of any number of lineages coalescing into one on the branch over the root of the species tree is always 1, because you have in some sense infinite amount of time. So to illustrate this, suppose this is a branch of the species tree. This is the length of the branch in terms of coalescent units. A coalescent unit is basically 2n generations, okay? So this is the length of the branch. And suppose I have this coalescent history, which means that I have 5 lineages entering the branch and 2 exiting, with 3 coalescent events happening in this specific order. So in that formula, the UB of H, for this specific coalescent history, the number of lineages entering here is 5. And the number of lineages exiting is, 2. So we have UB of H is 5, VB of H is 2. What is WB of H? What is the probability that I have 3 coalescent events will happen will happen to these 5 lineages that will give rise to exactly the species tree we observe? In this case, there are 3 of them. You could have even had the coalescent event 3 first, then 1, then 2, or 1, then 3, then 2, or 1, 2, and then 3. So these are the 3 possibilities, okay? Now, D sub B of H basically, this should be 6. Actually, it's 180. This is the number of all possible ways of 3 coalescent events happening on a set of 5 lineages in any order, even the orders that do not give rise to the same gene tree, okay? So here it's basically to choose the first coalescent event, we have to choose 2 out of 5. The second one, we have to choose 2 out of 4, because once we have a coalescent events, now it went down to 4, and then 3 choose 2, and so on. This will give 180, okay? And the formula for this actually is not bad. It's just the product of these terms here, where CB is the number of coalescent events that took place on the branch. Now, the probability of U lineages entering the branch and V exiting the branch within time t this is the term for it, and this was devised multiple times in the mid-80s. Okay, so with this, now this defines the whole framework for defining the probability of a gene tree given a species tree. Okay. Now that's the probabilistic setting, but we can use the notion of valid coalescent or coalescent histories also to define a parsimonious way for reconciling the gene trees. So in particular, Wayne Madison in 1997 proposed a criterion for reconciling gene trees that he called minimizing deep coalescences, or minimizing the number of extra lineages. So what is the number of extra lineages? Again, we have the gene tree that agrees with the species tree in both cases. I have 2 different coalescent histories here. If you look at each branch within the species tree, now you count how many lineages are there. and you subtract 1 from that, that will be the number of extra lineages. So how many extra lineages are still there? Because if everything under that branch has coalesced, you should see one lineage only. So anything beyond that is going to count as extra lineages. So in this case, we have 0 extra lineages here and 0, 0, 0. So there are 0 extra lineages over the entire tree. Here, A and B did not coalesce. So within this branch, we have one extra lineage. Okay, so one extra lineage here, 0, 0, 0, and 0 here. So overall, we have one extra lineage given this coalescent history. Now we can define this mathematically using the notion of coalescent histories, because now I can say mathematically that the number of extra lineages of a gene tree given a species tree is the minimum over all coalescent histories of the extra lineages given these specific valid coalescent histories. So you can go through every coalescent history, count the number of extra lineages for that coalescent history, and you take the minimum. Okay? The probabilistic setting said go over each one of them, compute the probability, and then sum the probabilities. Here we need to go over each one of them, do some simple counting, and take the minimum. Okay?
By the way, when you say number of extra lineages, this is summed over all of the edges in the tree.
Yes. Yes. Yeah. So now we have, using this concept of coalescent history, we have these two criteria for probabilistic way of computing the probability of a gene tree given a species tree. And we have this parsimonious concept of what is the minimum cost of reconciling a gene tree with a species tree. Now our problem of interest is we don't have a species tree. We have a set of gene trees. How do we get the species tree from that? So the problem is really a collection G of L gene trees, and we are interested in a species tree that optimizes certain criterion given that set G. Okay? So the issue here is that what kind of criteria? And there have been lots of them. I'm not going to go through all of them, but I will go through the probabilistic one and the parsimony one that are based on this concept of coalescent histories. So using this concept of coalescent histories and probabilities of gene trees, here's one likelihood setup for computing the species tree. So basically, we are interested in the species tree and its branch lengths that will maximize the product of the probabilities over all gene trees. So here, of course, the assumption is that the loci are independent, and therefore the probability of the set of gene trees is the product of the probability of each one of them. So now, under this case here, we can basically go through every possible species tree and every possible setting of branch lengths. For each one of them, we compute the probability of each gene tree under it, take the product, and then at the end, we take the probability, we take the species tree that maximizes that probability. These kind of calculations are implemented in the tool COL, which is implemented by James Dignan. The parsimony setup, MDC, is basically says, let's find the species tree that will minimize the sum of the extra lineages over all gene trees given a species tree. So go through every species tree, for each one, score the number of extra lineages of all gene trees, take the sum, and now return the tree that minimize that sum. Okay? So I will talk now a little bit about this MDC, which is, we have done some work on it, and I will start with the case of rooted binary gene trees. So how can we solve this problem if this input to the problem is rooted binary gene trees? And this is work I did with my student, and we published in 2009. So we basically have devised exact algorithms that do not explicitly try every species tree candidate. So you don't have to try every species tree candidate because that's ridiculous. And it doesn't even search the tree space. So we don't employ like SPR or TBR moves to go through the tree space and every time score it using the MDC criterion. The solutions, once you find that main insight there, the solutions are very simple. But there was an interesting insight that we got from there is that if you want to score if I'm given a collection of gene trees and I want to know the number of extra lineages that these gene trees will contribute to a single branch in the species tree, I need not know anything about the species tree other than that branch. So one of the main theorem in our paper was that the value of XLSTEGT, that if you give me a gene tree and you give me a branch E in the species tree, a branch, I don't need to know anything about the rest of the species tree. To compute the number of extra lineages that will arise from GT on branch E, it is independent of what other branches exist in ST, including the topology of the subtree under that branch. So if you tell me that this branch in the species tree has A, B, C, D under it, I can compute the number of extra lineages regardless of what is the structure of those A, B, C, D, what is the topology, okay? Now the interesting corollary that comes from that is that given a cluster W of taxa, so I want to check, does this cluster W, is it going to be in the species tree? So given a cluster W of taxa, now we can compute the number of extra lineages that would arise from reconciling all gene trees in G, had W been a cluster of the species tree. So now we can take for every cluster, every possible cluster, we can score the number of extra lineages that will arise if we put that W in the species tree. We will denote this number by K sub W of G. So K sub W of G is the number of extra lineages that will be on the branch over cluster W when we reconcile all the gene trees in G. And this is efficiently computable. Okay, it's computable actually in linear time in the number of leaves and in linear time in the number of gene trees. So what is the concept? Suppose I have this collection of gene trees. Again, this is part of the input. And suppose I want to know what would happen if someone tells me that this cluster is going to be in the species tree, A, B, C, E. To compute K sub W of G, all I need to do is find in these gene trees the maximal subtrees whose leaves are subsets of this. Okay. So if you find all the maximal subtrees whose leaves are subsets of this, it's this one, this one, and this one. These are the only maximal subtrees whose leaves are subsets of this. Now K sub W of G is this number of maximal subtrees minus the number of gene trees. Okay? So now I know that if you tell me that A, B, C, E is a cluster in the species tree, I can tell you that the number of extra lineages on that branch will be 2. For if this is the input of your gene, if this is the input gene trees. Okay. So now this gives rise to a nice computational framework for this problem, is that if you want to compute the species tree from a set of gene tree, you can get a set of all possible clusters of taxa. So for data sets with 10 taxa, try all 1,000 possible clusters. For each cluster W, use this technique of scoring it by K sub W of G minus the number of gene trees. Now it's a matter of just finding a maximal set of clusters whose total weight is smallest and whose elements are pairwise compatible. We want it smallest because we are minimizing the cost. We want it to be pairwise compatible because we want these clusters to fit in a tree. Okay. Now we have in that paper in 2009, we gave an integer linear programming approach for solving this problem, and we gave a dynamic programming approach. Both of them had very good results on synthetic and biological data set. And in addition there, we also proposed that, this you can imagine that the set of all possible clusters of this is 2 to the n, on the order of 2 to the n, where n is the number of leaves. So we proposed there also that if you are going to use thousands of loci or hundreds of loci, that probably you can focus on the clusters that are displayed by gene trees only. And that number is usually way much smaller than 2 to the n. So one of the heuristics we proposed there is to try only clusters displayed by gene trees. And we studied the performance of this heuristic. In most cases, we didn't see any loss, actually, using the heuristic. Sometimes, interestingly, actually focusing only on the clusters in the gene trees, you'll get even a better result. That's just an artifact of parsimony, okay? Not magic. I'm not going to talk about the synthetic data, results on synthetic data, because I don't have much time. But this is the result for this yeast data set that was proposed by Rokas et al. In 2003, where they basically sequenced 106 loci, sequenced these loci, concatenated the sequences, and built the species tree. They got this. The Bayesian approach, BEST, by Liang, Liu, and others, also got the same tree. Using MDC, we got the same tree. Of course, there's a huge difference in time between these two methods, but also BEST estimates other parameters like branch lengths, times, and population sizes, and so on. In addition, also, with our method, we also scored the suboptimal trees, because when we are dealing with parsimony, sometimes you have also to consider suboptimal trees to see what's the story there. And it was actually interesting because The optimal tree had 127. Well, the optimal tree will have 127 extra lineages once you reconcile all 106 gene trees. The suboptimal one wasn't much different, actually. It had 134, so it's just 7 extra lineages, 7 extra lineages to that one. And you'll notice actually that these 7 are caused here. And that was very interesting for us. Remember that the talk is about hybridization and so on, that when you look at these two trees, I mean, they are almost explaining the data almost equally well. The difference between them is that S-cud here, this Saccharomyces species, in this case, it is a sister taxa of S-bay. Here, it is a sister taxa of this. Now, if you think about reticulation and so on, looking at these trees, it will immediately tell you there's probably something about reticulation there. Now, the other ones here, these are suboptimal ones. The scores are getting much worse. Okay, I will get back to this data set later. So this is the case of when the gene trees are binary and rooted. Now, earlier this year, actually, with my student Yunyu and Tandy, this work appeared in Recom, and an extended version will appear in JCB. We focused on the case of when the gene trees may be unrooted, or may be non-binary, or both. Okay, and this is not just here's one more computational problem. Actually, this is the practical case of the problem. We need to deal with these cases because when you reconstruct gene trees, unless you have an outgroup or something like that, it's usually not easy to get a rooted gene tree. And for that, the non-binary gene trees is usually biologists, when they construct gene trees, they follow up with some sort of processing where they remove branches of low support. When you remove branches of low support, you will end up, or contract them, you'll end up with gene trees that are non-binary. So the question is, if you are given a set of gene trees that may be unrooted, maybe non-binary, or maybe both, unrooted and non-binary, how do you first define the MDC criterion? And second, how can you infer the species tree? You can imagine that this cannot be easier than the binary case. So our definitions of that, I will just talk about definitions here. Our definition on the unrooted gene tree is that if you give me the unrooted gene trees, what we are interested in is to find over all possible rootings of the gene trees. We need to go through every possible routing of gene trees and find the species tree that will optimize that MDC score. Okay, so here this is an illustration of given an unrooted gene tree and a rooted species tree. These are two possible ways of rooting. Now, usually the number of rooting is the number of ways of rooting a gene tree is the number of edges in the gene tree. But here I'm just showing two. If you root it like this, you will end up with 1 extra lineage. If you root it like this, you will end up with 3 extra lineages. So between these two routings, we will say that this is a better explanation. Okay. Now again, in theory, you don't have the species tree. In practice, actually, you don't have the species tree. You have a set of unrooted gene trees. You want, in theory, to look at every possible way of rooting each of them, and for each of them, then run our techniques to get the species tree. The case of non-binary gene tree. So suppose I have a non-binary gene tree. There is usually an exponential number of resolving the gene tree. It's exponential in the degree of the node. This is one way of resolving it. This is another way of resolving it. If you resolve it like this and score it, there is 0 extra lineages. If you resolve it like this, you get 3 extra lineages. We will say under MDC, because it's a parsimonious reconciliation, we would prefer this. Now again, we have to look at every possible resolution and then get the species tree from that. And the third case, of course, we combine the two. If the gene tree is unrooted and non-binary, now we have to root and refine, or root and refine and score. Okay. So for this problem, the input is a set of unrooted or non-binary or unrooted non-binary gene trees. The output is a species tree that minimizes XLST of G prime, which minimizes the number of extra lineages for given the G prime, which is basically the rooted version of G, or the refined version of G, or the rooted and refined version of G. Now the contributions that we give actually in the Recon paper and then in the JCB paper is to avoid this case here is that we don't want to try every possible way of routing. We don't want to try every possible way of refinement. I'm not saying there is an exponential number of routing a single gene tree, but if you look at k of them and you look, there is basically n to the k way, order of n to the k way to route them simultaneously. And there's an exponential number of refinements where it's exponential in the degree. And we don't want to do that because that will immediately make the method infeasible if the lack of resolution is very high in the gene trees. So the contribution of this paper was actually that we showed how to extend this main concept of k sub w of g to these cases. So now if you are given unrooted gene trees, we can compute the number of extra lineages over all possible resolutions implicitly. We don't have to resolve. We can actually compute it without resolving them. The same thing with routing, the same thing with routing and refining. Okay? And the method actually has been, these are all implemented. Tandy and her student actually have just recently submitted a paper on studying the performance of these methods when you do contraction of poorly supported branches and then reconstruction, and they show very good results. Actually, she has a copy of the paper if you're interested. Okay, so that actually, you're at half an hour. I finished talking about the gene tree, species tree. Now I want to move on to the case of what happens when we have reticulation. So this, all of this work here assumes that if you're given a set of gene trees and you want to reconcile them, you assume that the only cause of incongruence is incomplete lineage sorting. So now I want to talk about what happens if there is also hybridization in the picture. Of course, I am leaving out completely the issue of gene duplication and loss. It is extremely important, but I haven't done anything on it. This is why I'm leaving out. Not that it's not important. So, and that will be even much harder to deal with. I mean, to combine all of them. So basically, if we look at, there have been these two communities that have developed. There's one community that has developed a set of tools for gene tree reconciliation, assuming incomplete lineage sorting. You have BEST, BEAST, MDC, STEM, GLASS, all these kind of methods. And then you have reticulate evolution community, which has all these tools for inferring phylogenetic networks, assuming which, for this class of methods here, or in this community, basically the assumption is that all gene tree incongruence is due to reticulation. So usually, a parsimonious setting, for example, is to get a network with the minimum number of reticulation that will display all the gene trees in the input, or will display all the clusters displayed by gene trees in the input, and so on. Okay? So these two communities, again, focus, each one of them focuses on a single cause of incongruence and do reconciliation under that. But what we are interested in now is to look at both of them simultaneously. So in this case, for example, here's a phylogenetic network that describes the species. What this shows here is that, okay, you have sometimes hybridization, but in addition, sometimes also can get very, very complicated in the sense that if you trace the evolution of this lineage and this lineage from B and C, if this branch is too short here, these might not coalesce and they might take different paths. So now, given a gene tree, you have to account for the fact that things may not have coalesced and deep coalescence could have occurred, but also at the same time, you have to account for the case that hybridization could have occurred. So now we have these parameters that we have to worry about, T1 and T2, the length of these branches here, because this will affect the probability of deep coalescence. And we also have to have a new probability here, gamma or whatever, to account for the fact that if I am going backward in time here, what is the probability that I go left versus I go right? And under a likelihood setup, nothing much, I mean, it's almost the same, as before. The only difference is this n, is that now, if you're given a collection of gene trees and a phylogenetic network, the likelihood of the data and where the likelihood of the model, which is we need the branch lengths and the hybridization probabilities, is that now we can look again at the probabilities of the gene trees given a phylogenetic network along with its branch lengths and along with its hybridization probabilities. Okay, and we can take the probability, the product here. Now, we cannot apply that method that I described before for computing the probability of a gene tree given a species network, because now this is a different story. So given the main thing to be able to use this setup, the main question we have to consider is, given a phylogenetic network with its branch lengths, with its hybridization probabilities, and a gene tree, how do we compute this? How do we compute the probability of gene tree given that network? Now, Laura Kubatko has done some work on this, and this is a model that she and her student Meng here proposed. But the problem is very hard, and the models that have appeared are for very, very specific phylogenetic network. So these papers are not introducing here's how you do it for the general case. No, in this paper it was here is the phylogenetic network. There's two parameters in it. There's this branch length, and there is gamma here, which is the probability of going left versus 1 minus gamma, you go right. Now, if you have this setting, what they proposed here is that now you can compute the probability of a gene tree given this network by thinking about this network in terms of two trees that they call the parental species trees. So you can split this into these two trees that are the parental species trees, where B can go either left or right. You can now, the nice thing about this is now this probability of gene tree given the parental species tree and its branch lengths, the methods that we showed before applies directly to this and applies directly to that. The only difference now is that you have to multiply by gamma and 1 minus gamma. So now, given a gene tree, I ask what is the probability of gene tree given this phylogenetic network? This gene tree could have evolved down either this tree or this, which is the summation of these two probabilities. But if it evolved down this tree, we have to multiply it by gamma. If it evolved down this tree, we have to multiply by 1 minus gamma. Okay? So this was proposed in 2009. And they provided methods actually for the estimation of these parameters using maximum likelihood and Bayesian approach. And they obtained very good results. I mean, the parameter they were interested in, of course, is gamma, the rate of hybridization. And this is a table of results from their paper. They did simulation studies. These are the true values of gamma they used. This is the sample size here, the number of loci they used. And this is the maximum likelihood estimate of gamma. And you will see that actually the estimates are very, very close. Okay? So the performance was very good. But one of the issues with that is that this concept of parental species trees does not generalize beyond that simple model. Okay? And I'm not saying there was anything wrong with that paper, actually. It's just that that paper was for that specific model. And we thought about, can we take this model now and extend it? And the answer is no. You cannot extend this notion of parental species trees because, for example, if you have two alleles sampled from B, then the parental species trees will fail to account for all valid coalescent histories. So suppose I sample two alleles from B here. One can go this way, one can go this way. So if you look at this gene, if you look at the parental species trees, one of them forces both of them to go left, one of them forces both of them to go right. Neither of the two parental trees will allow you to model the case where this went left and this went right here. Okay, so if you have more than a single allele, that model of looking at parental species trees will not work. The other case, actually, you don't have even to invoke multiple alleles, that if you have hybridization and you have divergence, and you still have a single allele from here, then if you take one allele from here, one allele from here, again, you can go left and right at the same time for two different alleles, in which case that parental species tree where it's either B on the left or B on the right will not work. We ourselves try to fix that or try to generalize that to this case. So we thought about this case and we generalized it, and this appeared earlier in systematic biology earlier this year, where we allow for the case of divergence after hybridization. And this will account for the case of two alleles in the case of Laura Kubatko's model, and similarly with divergence after hybridization. And here the question was, again, how do we compute the probability of a gene tree for this model? And the only way to do it was explicitly, to actually compute the probability of a gene tree explicitly for each one of the gene trees. So here the way to read this is that the probability of each gene tree is the sum of these terms, each one multiplied by its coefficient here. So the probability of this gene tree is 0 times this, C times this, C times this. These A, B, C, D are basically functions of gamma, the hybridization probability. This G22 here, this is the PUV function from slides before. So Gij is the probability of I lineages coalescing into J lineages within certain time. Okay? And this will be, so actually you can do it for any phylogenetic network, but it is a very tedious process, and this would not generalize either. So These two approaches do not generalize. I mean, they are nice for these models and for other models that have similar properties, but still needed is a general network, a framework for computing the probability of a gene tree given any network N with its branch lengths and hybridization probabilities. And we have a solution actually that it is now in submission along with my student Yon and James Dignan also. Our solution is 3 steps. We convert the phylogenetic network into a multi-labeled tree, T. This is a very specific multi-labeled tree. It's not the way of looking at the gene trees being multi-labeled trees. We consider all allele mappings from the leaves of GT to the leaves of that multi-labeled tree. And then for each allele mapping, we compute the probability. And I will show them very briefly. So this is the pseudocode of an algorithm for taking a phylogenetic network compute and converting it into a multi-labeled tree. It's nothing actually hard. It's just that we take a phylogenetic network along with its branch length and hybridization probability. We need to get a multi-labeled tree. I will show, the description of it is here, but we will see it in a figure. We need to get the branch length, so we need to translate the branch length from the network to the multi-labeled tree. We need to translate the hybridization probabilities from the network to this. And we need to keep an edge mapping between the multi-labeled tree and the network. And let me show you an example. So if this is a phylogenetic network, here is a multi-labeled tree. It's very simple to convert it here. It's just you can go bottom up. Every time you hit a node of n degree 2, a node with 2 parents, just copy this whole species tree, create another copy, this subtree, and connect it once to the left parent, one to the right parent. Okay? This is very similar notion to the certain formats that were proposed for representing phylogenetic networks in UIC. Okay, so it's basically, think about this, that it can come one from here, one from here. So this will be the case. We have to translate again the branch lengths here. So this branch now that links U to W is now T8 plus T4. Okay, and gamma goes on this branch, 1 minus gamma goes on this branch. Now you can imagine if there's more hybridization here, that this number of B's and C's will start growing exponentially also. Okay, so this is how we go from phylogenetic network to multi-label tree. It's a very, very straightforward procedure. After we do that, now we have to find all allele mappings to that multi-label tree. What do I mean by that? Suppose we have a gene tree for which we have only a single allele from A, a single allele from C, a single allele from D, and 2 alleles from B. Now, if I give you this is the multi-label tree, then there are many possible ways of mapping these alleles to the leaves of the multi-label tree. Now, A and D, you don't have choices. A has to map to this A, D has to map to this D. But I have 1 allele from C, 2 from B. These 2 from B can map to this, C map to this. Or these 2 can map to this B and C map to this. You know, B1 here, B2 here, and C can go here, and so on. So you can compute this. And this is also, it's just, there's usually an exponential number of them, and you have to list them. Now, what we showed is that the probability of a gene tree given a network and its branch lengths and hybridization probabilities, it is really the summation over all possible allele mappings. For each one, we need to look at it, and then compute the probability of the gene tree given that multi-label trees with its branch lengths and gamma and that allele mapping. Okay? So what I mean by this is that you have to follow where you map the alleles to the leaves of the multi-label tree. So if you map them here, like under this allele mapping, there will be no coalescent events here, nothing here, nothing here. So there should be no probabilities corresponding to these branches because nothing was mapped to them. So for this allele mapping, all the probabilities have to take place, all the calculations have to take place on these branches here, but nothing on this. And if you do this, for example, B1 and B2 and C, if nothing coalesces here, then you have to multiply by gamma cubed here because 3 lineages went over that branch, and this is the probability of going that way. So now we reduce the problem from phylogenetic network back to tree, which is much nicer because every time we go back from the leaves to the root, there is a single path. Okay? But that left one piece of detail to figure out is how do you compute the probabilities given an allele mapping and a multi-label tree. It's not as simple as, okay, just run call, for example, and just do the regular calculations. Because now if you map B1 here and B2 here, These are not independent anymore. We have to be careful about when B1 goes here and B2 goes here. This amounts to, on the original network, to saying that B1 and B2 did not coalesce here, and you have to account for that. So if B1 goes here, B2 goes here, there is no way B1 and B2 could have coalesced on this branch. So we have to map this information, okay? This is how we map. I mean, I was told when I started graduate school that you can lose the audience with one slide, and here it is. Okay, I'm not gonna go through it, but the main idea here is that this notion here of Bn phi to the -1 of B prime is that we have to do the calculation, the product of all branches that map to a single branch in the phylogenetic network simultaneously. So if I go back to this, when I calculate probability on this branch and on this branch, I should not evaluate this and then this, or this and then this. I should wait. When I'm writing the algorithm, when I'm writing the program, when you compute the probability, remember that it is a product over all branches. But what we found, actually, our insight for this is that when you do that product, do not evaluate this and this until you have seen everything, and then take the product of these two simultaneously and evaluate it. So if you look at this formula here, this is very similar to what was before. It's the summation over all valid coalescent histories, but now we have also allele mappings. It is the product over all branches. Now this we have to account for gamma. If you have 3 lineages leaving that branch, you have to multiply by gamma over to the third, for example. Now we have this new probability, and this is just a symbolic term. It does not evaluate to anything. The reason it does not evaluate to anything is that to evaluate it, you have to find all the branches that map to a single branch in the phylogenetic network. Once you found them, their product will be this. Now this evaluates to something. Each one of these do not evaluate to anything, okay? So, and we have to account for the number of lineages entering a branch and leaving a branch in the multi-label tree. Again, we have to be careful about the mapping to the original network, okay? So this is the new framework, and now given any phylogenetic network with any scenario of hybridization, any number of lineages, and so on, it will be able to calculate the probabilities of gene trees. So we reanalyzed a yeast data set, and we considered 6 possible scenarios here. This is the species tree that's reported in every paper that analyzed it under incomplete lineage sorting. This is the second, the suboptimal one that we found under MDC that I showed. These 3 are phylogenetic networks that under MDC will explain the data best. So these are equally optimal under MDC. So we also extended the MDC criterion to networks. I did not talk about it here at all. But these 3 will be equally optimal for explaining the gene trees with hybridization. And this is the third one where we hypothesize that SCOD and SMEC have diverged after hybridization. For each one of them, we estimated the parameters, the branch lengths, and the value of gamma. For these 2, there is no gamma. For some of them, there is no T4. Some of them have T4, so we estimated that. For each one of them, we have the likelihood of the model. We have also 3 information criteria to account for parameterization of the model, because one of the things one has to be careful about phylogenetic networks is that add hybridization, you will fit the data better. Okay, so you cannot just keep adding and looking at improvement in the likelihood score, because by definition you will improve it. So here we accounted for that by using these 3 information criteria to look at how much improvement we get. And in fact, under parsimony, under MDC, you cannot distinguish between these 3. They are all equal. Each one of them invokes about 69 extra lineages, which is actually a huge improvement over this, because the number of extra lineages on this was 127. If you add 1 reticulation node, it drops to 69 immediately. Now with the likelihood, we find that this is the best scenario here, that SBay is really, there is hybridization between SCOD and something that either went extinct or not sampled in the data set. And the thing about it is that we estimate the hybridization probability to be about 34%. So that about 34% of lineages will follow this path and the rest will follow that path. And in terms of the information criteria, it was by far the best. I mean, it's much better than by just adding 1 parameter, it was much better than the best species tree. The other thing that's interesting about this is that the hybridization probability, the hybridization model, actually not only that said, of course, there is hybridization, but also it gave a very different estimate of this branch length here, the divergence times. So under species trees, you will get that, okay, T1 is short and T2 is short and so on. And you need to have that, actually. It makes sense that they are short because there was a lot of incomplete lineage sorting under assuming incomplete lineage sorting. But now, under this probabilistic setting, actually the model predicts that, no, there wasn't much lineage sorting. Actually, the branch was very long, and so much of the incongruence can be explained by hybridization, okay? And I have talked to certain biologists who have questioned incomplete lineage sorting, actually, and their reason for questioning it, they said that these branches are too long for incomplete lineage sorting to happen. And this actually supports that hypothesis that these branches are not as short as as these models would expect, but you will have to invoke some sort of hybridization here. We also looked at synthetic data. For example, we took this phylogenetic network. We evolved gene trees down this one with varying values of alpha, beta, T1, T2, and T3. In this case here, we are showing the performance of the method in estimating alpha and beta once you fix T1, T2, T3. So if we tell the method, here are the true values of T1, T2, and T3, estimate alpha and beta, it does very well, actually. And the x-axis, it shows as the number of gene trees increases. On the y-axis, this is the probabilities that we estimate, or the values of alpha and beta. Here, in this figure, we use a true value of alpha equals 0 and beta 0.5. And you see that alpha is getting to 0, and this beta is going to 0.5. Here, we use 0.3, 0.3. We are getting good estimate. Here also 0.5 and 1, we are getting good estimate. Now, here we assume you know T1, T2, T3. If you don't assume them, we show that you get non-identifiability. You cannot identify them. You can find so many values that will give exactly the same probability. But there is no magic here, because if you look at just T1, if you try to estimate T1, T2, T3, alpha, and beta, there are 5 parameters to estimate, and you have only 3 gene tree possible. There is A, B versus C, A, C versus B, and B, C versus A. So you have basically so many equations, but only 3 variables, okay? Now the other part of the figure shows identifiabilities. Assume that alpha is 0. So suppose alpha is 0, so now all your genetic material is coming from here. If we have alpha 0, now we look at this. So suppose I have this scenario with alpha 0 and beta can be whatever, and we have this competing scenario. We find actually that no matter what you set T2, T3, and beta to, you can always find a setting for T that will give the same likelihood or probability of the gene trees under this model than the same thing with his. So what this means is that if your phylogenetic network model was really one hybridization event here with this probability beta, there was an extinction here, and you try to estimate T2 and T3. T1 is irrelevant because there's a single allele. If you try to estimate T2, T3, and beta, whatever values you get, I can show you another phylogenetic tree with a different value of T that will give the same probability to your gene trees. And these are actually some figures that show that if you try alpha is 0, beta is 0.1, and this is T as a function of T2 and T3. So you change T2, you increase T2, you increase T3, you just increase T and you will get it. In fact, actually it's easy to get an exact equation for this. So whatever value for beta and T2, T3, if you set T to this value, you will get, no matter how large your gene tree set is, you'll get exactly the same probability. Okay, so using this framework, we can infer species phylogenies that account for incomplete lineage sorting and hybridization. We can look at identifiability questions and so on. So to summarize, the concept of a coalescent histories is central to defining reconciliation criteria and computing with them. Efficient solutions for MDC criterion on trees, we have proposed them for when the gene trees are binary, non-binary, rooted, unrooted, with single alleles, multiple alleles, all of that we have solutions. We have a general method for computing the probability of a gene tree given a phylogenetic network. We show its application to detecting hybridization and incomplete lineage sorting. We also have the MDC criterion for networks, and again, there's no way I could have shown it. All these methods, even MDC on networks, enumerating valid coalescent histories, all of that, they are implemented in a software package called Phylogenetic, which is available here. So this work was done in collaboration with James Degnan, Quang Tan, my former student, Tandy, and my current student UNU. I want to acknowledge the funding agencies, and I definitely want to thank you for your attention.
We have time for some questions.
So, very interesting. I love it. But one thing I wonder about is it seems the structure where you take the gene trees as given and then look at species tree hybridization reconstruction, is it removes the possibility that that structure of having a shared species tree can inform better estimates of the individual gene trees.
So yeah, definitely. I mean, there are methods. So when I, for example, talked about BEST, for the yeast data set, MDC infers the species tree in fractions of, like a few seconds. BEST takes 800 hours. And the reason one of the reasons for that is also that BEST goes from the sequence alignments, right? So they try to infer the gene trees and the parameters and the species tree simultaneously. So yes, that is definitely an issue. Here, the assumption here is that the gene trees are independent. The assumption also is that let the user build the gene trees with whatever method they want, and then let's infer the species tree. Now, if there was some sort of correlation across the gene trees, definitely this method might start suffering, right? Because you might start missing that information among, in the lack of independence across loci, right? So, I'm sure one can design a simulation study and put all that, correlations among the gene trees that might actually not favor this kind of setting.
I guess, sorry if I could just follow that up. Do your methods allow for a set of potential gene trees for each gene? If there were some other method used that estimated an uncertain set?
So one of the implementation here is where you use an unresolved gene tree. So if you have a set of gene trees for a single locus, in current implementation, one way to do it is do some sort of summary of this set of gene trees, take majority consensus, strict consensus, whatever, and then apply this. Then the nice thing about it is that it's going to resolve it in such a way that will optimize the criterion. Now, if you give me a set of gene trees and you still want to use them to analyze each one of them independently, it's just a matter of wrapping a script around this and doing it. It's just going to be a lot of time, right? Because if each set because we have to try every combination, and that's going to be exponential.
One more question.
I don't know. There are 5 hands there. I don't know which one.
Choose.
You choose. You
choose.
are my advisor.
Okay, I really like your talk, but I'm concerned about the choice of the data that you use to illustrate it with.
What, the synthetic data or the biological?
No, the biological data. People have already published papers on this data set suggesting that the sequences are not evolving under globally stationary, reversible, and homogeneous conditions. I played around with it as well, haven't published it yet, but it's absolutely clear that the evolutionary process over that data has shifted over the whole tree gradually, along one edge to the next stage, to the next stage, to the next stage. You have a different Markov process along each edge. And so, obviously, by breaking up into smaller data set, the power, or smaller genes, the power of any test that you do is minimized, but you also are able to detect bigger differences there are in between the genes. So it might in fact be that the differences between the genes trees that you get actually is not a representation of what you think it is, but it's actually a representation of a shift in the evolutionary process over the whole tree for each individual gene. Have you thought of that?
Clearly not.
Okay, I think you can take that out over a coffee later,
Yeah.
because now it's time to go to the next speaker.
Yeah, I'll be happy to talk to you about that.